We're going to start the next part of our DNA section with a really important part of the process, and the reason why we even have DNA, and that is making proteins. The whole point of DNA is it's a cookbook, and I've said that several times throughout the section, but it's not a cookbook for making cookies or anything else like that. It's a cookbook for making proteins, and we use proteins in pretty much every part of our living organism, right? Doesn't matter what we're talking about. We could be talking about our bones, our muscles, the antibodies that fight off disease, right? You're hearing about this vaccine. Everyone's getting a vaccine so we can build antibodies. These are all proteins. So how do we go from DNA to an actual protein? And that's what we're going to talk about in this section. So I want to first just do a quick review, a little bit about the cell itself. So quick review of cell biology. So in a cell, and we're talking about eukaryotic organisms. So these are organisms that have a nucleus. This process does happen in prokaryotic organisms. It's just not this complex. So we're going to go through the eukaryotic process. So in here we have our DNA, and our DNA is in the nucleus. Now the DNA cannot leave the nucleus for several reasons. So why can't the DNA leave the nucleus? Well, first off, it can't leave the nucleus because it's too big. It is a double-stranded molecule. It is really long, and it's just a large molecule. So imagine a doorway that's too small to get out of. You might want to get out of there, but you can't. The other reason why it doesn't leave the nucleus is it's protected there. This is our master copy of the cookbook to make every protein in a living organism. And if it gets affected at all, it gets mutated. And what can a mutation mean? Well, that can mean something as simple as nothing can happen. It could be a very simple mutation, or it could cause cancer. So we want to protect this DNA so it cannot leave because of that. So because the DNA can't leave, we need to figure out some way to get the information from the DNA out of the nucleus. And that's what we're going to talk about today. How do we actually get this information from the nucleus out to where it needs to be made? So let's talk a little more about what's going on in my little cell drawing. I drew a couple organelles. So we have our nucleus. Here we have our rough ER and our smooth ER. And if you remember back to that section a couple sections ago, the ER, or the endoplasmic reticulum, is there for the transport of proteins. So as the proteins get made, the endoplasmic reticulum is essentially this pathways, set of pathways which are going to be used to send things around the cell. And then down here we have our Golgi body. And the Golgi body has a really important job. That packages the proteins that get made inside the cell before they go out. And then this last thing I drew on here are these little purple dots, and those represent ribosomes. And the ribosomes aren't just on the rough endoplasmic reticulum. That's why it's called rough, because it has those ribosomes. The ribosomes are also found in the cytoplasm. So I should have probably drawn some of them out in the cytoplasm. So I'll add some ribosomes out here. They are just kind of hanging around in the cytoplasm. So what ends up happening here is the information that is in the DNA, we need some way to get that information out of the nucleus to the ribosomes so the ribosomes can make the proteins. When the ribosomes make the protein, so let's say this is the protein that gets made. Make it a different color. So let's say this is the protein here, and that protein gets made. Well, now I need some way to get this protein out of the cell. So the protein gets made, it then gets transported out of the endoplasmic reticulum, and maybe it gets used in the cell. This might be a protein that gets used inside the cell, and it gets used right away. Or it goes through the Golgi body, and the Golgi body is then going to package it and into its protein coat, and then it's going to be able to leave the cell and go out and do whatever it does. So maybe it's making a hormone that gets secreted from the cell. Maybe it's making a neurotransmitter. Maybe it's making antibodies, but it's going to make something and send it out to where it needs to go. So what's going to actually copy that DNA? What's going to take the DNA from the nucleus and bring it out to the ribosome? That's going to be what we're talking about today, and that is a very important molecule called RNA, or ribonucleic acid. So this is the new bit of information from the last section. So we have DNA, which stands for deoxyribonucleic acid, and now we have RNA, and RNA is ribonucleic acid. It is a very similar molecule to DNA, but its job is very simple. Its job is to copy the DNA from the nucleus and bring it to the ribosomes. The DNA cannot leave the nucleus, so it needs some way to get that information to the ribosomes, which are going to be out on the endoplasmic reticulum, and they're going to be out floating around in the cytoplasm. So imagine I can't leave my room right now. I'm stuck here. I can't get out. And I need to get some information somewhere outside of my house. I might ask somebody to help me out. I'll call a friend over and say, hey, I'm going to write down this message. Do me a favor. Bring it to where it needs to go. So the whole point of this process is to make proteins, right? I can't make proteins in the nucleus. If I make proteins in the nucleus, they're going to be too big to get out. You can't, it would be like making, it would be like if I made cars and I tried to build a car in my office instead of on the factory floor. I might be able to build that car in my office, but once that car is done, I can't get it out of my office. So I need to build these proteins where it makes sense to have them. So, you know, if I go back to my diagram here, just one more time, the proteins get made out here so they can get used outside of the cell. If I made the proteins in here, they would not be able to get out of the nucleus. So I need to make them out here where they can be used and sent out to where they need to go. All right, let's go a little farther into a little bit about our friend, our new friend. This is our new friend, ribonucleic acid, or RNA. So a couple things about RNA. It is a messenger, right? And there's actually 3 types. So the first type I'm talking about here is the messenger. It copies the information from the DNA and it brings it out to the ribosomes where it's going to actually get used. It would be like if I'm a cook, right? Or I have a recipe I want to make. I don't have a kitchen here. I can't make the food right here. So I might copy down the recipe and give it to somebody to bring to the kitchen to make it there. So what are some structural components about ribosomes? Well, for one thing, it doesn't have the sugar deoxyribose. So in DNA, if you remember back to DNA, it's got 3 components to it. It's got a phosphate, it's got the sugar, which is deoxyribose, and then it has one of the 4 bases, A, C, T, and G. Now, RNA is almost the same, except instead of deoxyribose, it's got a little bit of a different sugar. It's got a sugar called ribose. So it does the same job. It's just the structural component. It's what holds onto the base, but it's going to be as ribose instead of deoxyribose. The next big difference, and this is important, is that RNA is single-stranded, not double-stranded. So why is it single-stranded? Well, it's just copying the DNA, so it doesn't need to be double-stranded. And also, it needs to be able to fit out of the nucleus. So if you take a look here, this is DNA in this molecule. It's a double-stranded molecule. And then RNA is a single-stranded molecule. So in order to get out of the nucleus, it has to fit out of the nuclear pores, these little openings in the nucleus. The DNA is too big to do that, but the RNA is not. So being single-stranded allows that RNA to get around the cell the way that it needs to. So the second big difference between DNA and RNA is that DNA is double-stranded and RNA is single-stranded. And then the final big difference, now there's more than just these 3, but these are the big differences, is that there's no thymine in RNA. There's another molecule, there's another base there, it's called uracil. So it does the same thing as thymine. It pairs with adenine. So adenine pairs with thymine, but in RNA there is no thymine. Adenine pairs with uracil. So let me give you an analogy. If I go down to Philly right now and I want to get a sandwich on a long roll with lettuce, tomato, onions, Italian meats, and cheese, what am I going to ask for? I'm going to ask for a hoagie, right? So around here, everybody knows what a hoagie is. If I go up to New York, where I'm from, and I order a hoagie, they're going to look at me like I'm crazy. That's not that far away, but they're going to be like, what? Even though it's just an hour and a half away, they're going to say, oh, you mean a sub? And then if I go out to Pittsburgh, they might call it something else. I think they call it a wedge up there. Some places they call them heroes. The point I'm trying to make here is a long sandwich is a long sandwich. But depending on where you go in the country, they call it something different. So uracil is kind of like that. It's a molecule that does the same thing as adenine, but it pairs up differently. So let me just explain that a little bit better with an example. If this is my strand of DNA here, right? So I have A, C, G, T, T, A, G, A. Now let's say I want to copy this. I want to make an RNA strand. So what ends up happening, and I'll explain how it does this later. We don't have to worry about how it does it yet. But I just want to show you how it would pair up. So I would have a new strand forming, and now we're making our RNA. But there's going to be a little bit of a difference. C is going to pair with G like normal because there's no difference there. So I'm going to do my C's and G's first. But now, if you remember, there's no thymine in RNA. Thymine is replaced by uracil. So A is not going to pair with T. A is going to pair with U. T is still going to pair with A because adenine exists, but there's no thymine. So A is going to pair with U, and A is going to pair with U. So this is our RNA here, and then this would be our DNA here. So another big difference is that DNA has thymine and RNA has adenine. So just a quick review of what I went over. So in terms of the differences between DNA and RNA, DNA is double-stranded and RNA is single-stranded. That's a big difference. Another big difference is that there's a sugar called ribose. So if this is DNA, it will be phosphate, deoxyribose, and a base, right? We've been talking about this a bunch, and that's going to be the base. Now, RNA is the same exact thing, but RNA is going to have a phosphate attached to a little bit of a different sugar called ribose, and then that's going to be the base. So they look almost the same, but the difference is that ribose sugar. And then finally, we have our uracil instead of thymine in our RNA. So just to finish up this lecture, in order for the cell to make proteins, we need some way to get the information from the DNA that's in the nucleus out of the nucleus out to where it gets made in the cytoplasm. And that is solved by this very important molecule called RNA. And RNA is just a tool to copy the DNA so it can be taken to the place where the proteins actually get made. And that's what we're going to talk about in the next lecture. All right, how do we actually read this DNA and copy it? So thank you very much.